Welcome back to the SANS Science Experience, where we explore the most exciting new research papers and preprints just for you.
Great to be back.
And hey, if you're enjoying these deep dives, finding them useful or just plain interesting, please do hit that like button and subscribe. It genuinely helps us keep making more.
It really does.
So today, we're getting into the secret lives of mitochondria.
Ah, yes. Everyone knows them, right? The powerhouses of the cell, making all that ATP energy.
Exactly. That's the textbook picture. Energy factories. But it turns out that's maybe not their whole story. They're incredibly flexible.
Super adaptable. And they've taken some really, really surprising evolutionary paths.
It's like a story of cellular renovation, you could say, where these mitochondria have sometimes totally changed jobs, evolving into things called mitochondrion-related organelles, MROs.
MROs, yeah. And to really get a handle on this flexibility, we're unpacking a pretty recent study that uses some clever computational approaches.
Right, so using computers to track evolution.
Exactly. It looks across, pretty much all eukaryotes, everything from yeast to us, to try and figure out how and maybe why mitochondria have changed so dramatically in different life forms.
And this isn't just a few weird cases, right? This is like a big pattern.
It's widespread, definitely. The researchers used a method, they call it hypercubic inference.
Hypercubic inference.
Yeah.
Okay, let's break that down a bit. Sounds intense.
Well, think of it like this. You have a set of features mitochondria can have, like switches. This method tries to figure out the most probable order those switches got flipped off during evolution.
Ah, okay. So which bits disappeared first, which ones later, to get the different kinds of mitochondria and these MROs we see today.
Precisely. And the study focused on a key set of these features, these switches.
Which ones were they looking at?
Okay, so they included the TCA cycle, that's central metabolic roundabout in the cell.
Right.
Then the main energy-making parts. Complexes I through 5e of the electron transport chain.
The big players for ATP.
Yeah. And ATP synthase itself, that molecular turbine.
Right.
Also the mitochondrion's own DNA, which is often lost.
mtDNA.
Pyruvate dehydrogenase, or PDH, which links sugar breakdown to the TCA cycle. And finally, the machinery for making iron-sulfur clusters.
Those little helper molecules, FES clusters.
Crucial helper molecules. And what's really striking, as you hinted, is that this iron-sulfur cluster machinery seems to stick around. It's often the very last thing to go, even in the most stripped down MROs.
Which strongly suggests it's doing something absolutely critical, even if making ATP isn't the main point anymore.
Exactly. Which leads us to, what are these MROs, more specifically?
Good question. Let's zoom in.
So think of MROs as mitochondria that have gone through some serious downsizing. They've lost some, maybe even most, of their original toolkit.
Streamlined versions?
Yeah. A classic example is the hydrogenosome. You find these in some anaerobic microbes, things that live without oxygen.
Okay.
They can actually still produce a bit of ATP, interestingly, but they kick out hydrogen gas as a waste product.
Hydrogen. Okay, that's different. What else?
Then you've got mitosomes. These are even more reduced. They're like the minimalist version. They've completely lost the ability to make ATP.
Wow, mitosomes. So mitochondria that have totally retired from the energy business.
Pretty much. And the study even mentions different classes, like class I to V, based on what metabolic jobs they still do or don't do.
So it's a whole spectrum of reduction.
It is. And here's where it gets really cool, I think. This whole process, mitochondria changing, losing bits, becoming MROs, it hasn't just happened once. It's happened over and over and over again, independently, in completely different branches of the eukaryotic tree of life.
Wow, okay, so that's convergent evolution, right?
Textbook convergent evolution. Different organisms, different starting points, maybe similar environmental challenges, and they arrive at similar solutions.
Yeah.
These modified, reduced mitochondria.
Different paths leading to kind of similar endpoints.
Fascinating. Okay, so how did the researchers actually map out these different evolutionary roads? Back to the hypercubic inference. Right, the hypertrapes method they used. What it does is it looks at which organisms have which of those features, TCA, complex I, DNA, FES, etc., across a huge family tree of eukaryotes.
Okay, using the tree of life.
Yeah, and it tries to reconstruct what the ancestral mitochondria might have had. Then it calculates the most likely sequence of losses that connect that ancestor to what we see today. It's like reading the evolutionary history book of mitochondria.
Like watching evolution in reverse for these organelles. Amazing. And a big finding was that even with all this variety, it seems most MROs followed one of just two main routes.
That's the core finding, yeah. Two dominant patterns or pathways of losing features. They call them the CI class pathway and the alt class pathway.
CI class and alt class. And the names relate to what goes first.
Exactly. The CI class pathway usually, or often, kicks off with the loss of complex 1.
Okay, complex 1 is the first domino in the CI class. What usually happens after that?
The model suggests that after CI goes, complex 3 is often the next major loss. Then frequently see complex 4 in the TCA cycle disappearing. ATP synthase and mitochondrial DNA, they tend to go later in this pathway.
Right.
Followed usually by complex 2 and pyruvate dehydrogenase, sort of towards the end game. Although they note PDH loss can sometimes happen earlier in this CI class route.
There's some variation there.
A bit of variation, yeah. And then, almost always last, is that FES cluster machinery.
Okay, so that's the general blueprint for the CI class breakdown. How's the alt class pathway different? Where does it start?
The alt class pathway, it starts differently. It usually begins with losing complexes 3 and 4, and often the TCA cycle goes early too, alongside them.
So CIV, CIV, and TCA tend to vanish together early on.
Yeah, relatively early, and often seemingly linked in this pathway. After that group is gone, the typical sequence seems to be losing ATP synthase and the DNA.
CV and mtDNA.
Right, then PDH, then complex 2, then complex I actually gets lost quite late in this pathway.
Interesting. So CI is late loss in the alt class.
Exactly. And again, FES assembly is usually the last one standing.
So the very first steps really seem to set the trajectory. It's like choosing a path at a fork in the road.
It really looks that way.
And the research also looked at interactions, right? Like, does losing one thing make losing something else more or less likely?
Absolutely. That's another powerful thing hypertrapias can infer. They found evidence for these kinds of influences. For instance, losing complex 4 seems to make losing complex 3 more probable.
Okay, which fits that early CIA-CIV co-loss in the alt class path.
Precisely. And similarly, losing the TCA cycle makes losing complex 4 more likely, again, supporting that alt class initial pattern.
So losses can encourage other specific losses down the same road, where there are inhibitory effects too, where losing one thing made another loss less likely.
Yes, interestingly, they found that losing complex 4 actually seems to make losing complex I less likely.
So that helps keep the two pathways distinct. If you start down the alt path by losing CIV, you're discouraged from immediately losing CI.
That seems to be the inference, yeah. It helps maintain those two separate routes. They also saw something they described as like a mutual repression, a kind of seesaw between ATP synthase CV and pyruvate dehydrogenase, PDH.
The seesaw. How so?
It might help explain why PDH loss timing varies in the CI class pathway. Perhaps losing PDH is less favorable or maybe even detrimental, while the mitochondrion is still heavily reliant on ATP synthase for energy.
That makes sense. The dependencies change as the system breaks down. It's fascinating how interconnected it all is, evolutionarily speaking. Did they look at specific groups, like did certain organisms prefer one path?
They did. They looked at specific clades. Ciliates, for example, a really diverse group of protists, they seem to predominantly use the alt class pathway.
Mostly alt class for ciliates.
Yeah, and losing complexans completely seems less common for them. But then you look at apicomplexans.
Those are mostly parasites, right? Like the malaria parasite.
Exactly. Apicomplexans show a really strong bias towards the CI class pathway, but with a twist.
A twist.
In their version of the CI class pathway, pyruvate dehydrogenase, PDH, tends to be lost very early, like one of the first things to go right after or even alongside complex I.
Wow, so they have a highly specific, almost canalized version of the CI class route.
It seems very constrained, yes. Their evolutionary path in this respect looks quite set.
So the organism's lifestyle, maybe being a parasite, could really push evolution down one specific track. What about the speed of these losses? Did they get any sense of timing?
They did try to estimate timescales using molecular clock data for some lineages. It's tricky, and the estimates have wide error bars, as you'd expect.
Sure, deep time estimates are hard.
But there was a general trend. The losses in the alt class pathways seem to happen on somewhat shorter timescales, generally, compared to the CI class losses.
So the alt path might be faster once it gets going.
Potentially, yes. Especially that initial loss of CIV, CIV, and TCA that seemed to happen in relatively rapid succession in many cases following that path.
Like a quicker dismantling once those key parts are gone. Okay, the final piece was this computer model, MitoMammal. How did simulating mitochondrial metabolism help explain these pathways?
Right. MitoMammal is basically a detailed simulation of how specifically mammalian mitochondria function metabolically. They used it to ask, what's the impact on ATP production if we hypothetically remove component X or component Y?
Like a virtual experiment.
Exactly. And the results were really insightful. They found, for example, that losing just complex I on its own actually has a relatively smaller impact on the maximum ATP production rate compared to losing complex 3 or complex 4.
Huh. I might have guessed CI loss would be catastrophic.
Well, it has an impact, but perhaps less than CI or CIV, especially if the cell doesn't have ways to compensate. They also found losing PDH alone had a pretty limited effect on overall ATP output in the model.
Okay.
But as you'd expect, knocking out the entire TCA cycle, that significantly tanks ATP production. Though even within the TCA cycle, losing different enzymes had varying impacts.
Right, not all steps are equal. So how did this simulation link back to the CI class and alt class pathways they observed in real organisms?
Here's the really neat connection. The model suggested that once you lose complex I plus any one of CIV or the TCA cycle, well, at that point, the potential for ATP production drops close to a minimum.
So once that initial hit happens, CI plus one other key part, the energy gain from keeping the rest of CI, CIV, TCA might be marginal.
Exactly. From purely an ATP perspective, there might be little selective advantage to retaining the remaining bits of that core machinery once CI and one of the others are gone. This could explain why they tend to be lost relatively quickly together in the observed pathways.
It lowers the pressure to keep them.
Precisely. And more broadly, they saw a correlation. Features whose loss caused a bigger hit to ATP production in the model were generally less likely to be lost early in the inferred evolutionary pathways.
So the metabolic cost seems to directly shape the evolutionary trajectory, the path of least metabolic resistance, in a way.
It strongly suggests that, yes, the metabolic consequences seem to be a major driver sculpting these evolutionary routes.
Wow. Okay, this has been a seriously fascinating look at how flexible mitochondria really are. Much more than just power plants.
It really has. We've seen that they've largely evolved along these two main routes, CI class and alt class, probably shaped by the metabolic impact of those initial losses, and that different groups can favor different paths.
And it really shows the power of using these computational methods like hypercubic inference to piece together these incredibly complex ancient evolutionary stories for the patterns we see today.
Absolutely. It's a great example of how computational biology is letting us tackle these huge evolutionary questions.
So here's a thought to leave everyone with. Given that iron-sulfur cluster assembly seems so vital, so stubbornly kept even when ATP production is gone, what other essential jobs, completely unrelated to energy, might these MROs be doing? What are the secret functions driving the retention or loss of other parts in different organisms? What are they really up to?
That's the million-dollar question, isn't it? What else are they doing? A great point to ponder.
Definitely something to think about. Well, thanks again for joining us for this deep dive into mitochondrial evolution. If you found this as interesting as we did, please remember to hit like and subscribe. It helps us bring more science stories to you.
Thanks for listening.